Okay, awesome. Hi, everyone. This is the last Sybeco of the year. Today, we're hosting Dr. Xiyu Zhu. He's a postdoctoral fellow in the lab of Dr. Vikaas Sohal at UCSF. His research focuses on the prefrontal cortex and how it supports adaptive behavior with implications for psychiatric disorders. Xiyu did both his bachelor's and PhD at the University of Pittsburgh. In his undergraduate work, which he did in the lab of Dr. Colin McClellan, he studied the molecular basis of circadian rhythms. And in his PhD in the lab of Anthony Grace, he investigated the midbrain dopamine neurons and their role in schizophrenia. Xiyu, broadly speaking, is interested in understanding how distributed neural circuits regulate flexible behavior and how disruption of these mechanisms contributes to psychiatric conditions. Welcome. Go ahead, please. You're muted. Sorry. Go ahead. Yeah.
Hi everyone. I'm so sorry. I apologize for the delay. I will rush through the background and the methods section and to hopefully catch up on the missing time. My name is Xiyu, and I'm a postdoc member in the Vikaas Sohal lab. Today, I'm going to present on our most recent preprint on synaptic plasticity of prefrontal long-range PV interneuron and how that regulate cognitive flexibility behavior. So here is the overview of today's talk. So first, I will give a brief background and on the significance of why we are interested in this long-range circuit in the PV neurons. And then I will bring us to the research questions we are interested in. And I will also talk about the methodology, followed up by the results and the discussion for this paper. So first of all, we are interested in cognitive flexibility because this is a fundamental ability of any organism and allows the organism to switch strategies and attention when prior learned association apply no longer. It is a cognitive flexibility is a core function of executive function, which is largely mediated by PFC. Cognitive flexibility and the impairment in this function is a hallmark in several psychiatric disorders, ranging from schizophrenia to autism to addiction. And it is known that the PFC and its downstream structures, such as the thalamus and the striatum, can coordinate their activity and activity patterns to adjust the behavior, allowing real-time update and to allow flexibility. And it is also known that this diverse class of interneurons in these structures is required for cognitive flexibility to occur. Therefore, knowing what is a neural substrates of cognitive flexibility underlying PFC circuit remains great translational value. However, although this circuit in the PFC thalamus and striatum is important, the mechanistic insight is still poorly understood. Precisely how does brain abandon previously learned rule and representation and implement new ones is unclear. Like I mentioned in the previous slide, the medial prefrontal cortex in the rodent brain is known to support flexible behavior, allowing a mouse to adapt to a new context. And it is known that if you, both the pyramidal neurons and the interneurons in these regions are important. And it is known that especially inhibiting the most abundant interneuron subtype, parvalbumin interneurons, can drastically alter flexible behavior. For example, in Callan-Dunk lab at Columbia University, they demonstrated that silencing PV interneurons during a task that allows cognitive flexibility, one can make the mouse more difficult to learn a new association upon the previous learned association in the digging-based cognitive flexibility task. And in our lab, we did a similar experiment, but inhibiting all the GABAergic neurons in the PFC during a rule shift task. I will delve into more details of this task in a later slide, but we can also find that the mouse spent more trials to learn the shifted rule, and the mouse also make more preservative errors during this process. And this on the contrary, people also find that by activating prefrontal PV interneurons, one can rescue cognitive flexibility deficit in diverse rodent models related to schizophrenia and interneuron impairment. This include MKA01 model, the DLX56 model of interneuron deficit. Although these broad behavioral phenomena were known, it is still unknown what are the important synaptic substrates in the PV circuitry that's driving this effect. So in our lab, we are interested in long-range PV inhibition in the prefrontal cortex and their role in cognitive flexibility behaviors. Starting 2015, we have published 3 papers looking at this specific projection coming from one hemisphere of the PFC and to the contralateral hemisphere. So specifically in 2015, we published a paper demonstrating that by by stimulating PV interneurons in the PFC at 40 hertz, we can rescue a cognitive flexibility deficit in the DLX56 model of interneuron impairment. This broadly implicates that the global dysfunction of medial prefrontal cortex, all the interneuron can rescue and impair flexibility. However, one question still remains, which is like what cell type and which circuit and what neurodynamics are required for this phenomenon. Oh,
Question there. So with the when you say long range inhibition, I guess so is it, so we have some local PV interneurons, and then we have some PV neurons that project from one hemisphere, you said, to the other hemisphere?
Yes, yes. So this is a recently found circuit from our lab, but in sensory cortices such as from the auditory cortex, the one hemisphere of auditory cortex to the other, there's also a sparse projection of PV and also of SST. And in our lab, we also find that from the prefrontal cortex to the hippocampus, there are GABAergic connections.
And these are different subtypes of PV and SST neurons?
So I can speak for the PFC to hippocampal connections. So the majority of them are PV, but there are also GABAergic connections which we haven't really studied. But so the majority of the connections they are PV.
Okay, thanks.
Yeah, sure. So follow up to our 2015 paper where we demonstrated that the global dysfunction in the medial prefrontal cortex interneuron long-range projection. In this paper, we focus specifically on the colossal long-range PV connection. And we find that during a version of cognitive flexibility task, the rule shift task, when animal make an error, their cross-hemispheric gamma synchrony, which is the gamma synchrony within the PV, the left and the right PV neurons, they emerge when animal just made a mistake. And also behaviorally, what's interesting is that we find that if you give animal out of phase 40 hertz stimulation in the PFC, you can impair rule shifting performance. This further indicate that it's not just that the transhemispheric GABAergic connection is important. It is the PV interhemispheric connection that's driving the PV gamma synchrony to emerge during the flexible behavior. And also this connection is important in forming a activity pattern in the interhemispheric interneuron connection that's facilitating rule shifting behavior. So following up to the 2020 paper, we next try to identify an anatomical connection, a synaptic connection, which can explain all this.
Questions you. So about this gamma synchrony. So we see gamma mostly as like thalamocortical bottom-up sensory inputs, right? So when you say PV gamma, like is the idea that, yeah, I'm curious, where is this gamma coming from?
We use voltage indicator recording in our 2020 paper. So basically we find that the gamma rhythm, so the gamma synchrony is within PV interneurons themselves. So basically, we find that at a population level, the PV, so it's not like LFP recording. So we looked at cell type specific synchrony of the PV interneuron in one hemisphere and how that synchronized with the rhythm of PV interneuron on the contralateral hemisphere. So that activity emerged when animal make a mistake in rule shifting task.
So the gamma emerges when they make mistakes.
Yes. So they all emerge when they make mistake, and it remain relatively stable when they make correct choices.
I see. Okay.
Right. So in our 2023 paper, we identified an anatomical connection originating from PV in one hemisphere. And these neurons do send long-range connection monosynaptic to the contralateral hemisphere. And we find that if you only manipulate, if you only inhibit those long-range PV terminals, you can impair rule shifting performance. And what I didn't show here is that this behavioral change is actually reversible. On the next day, if you give this projection 40 hertz stimulation, you can rescue this behavior. And we also have an experiment demonstrating that both the impairment and also the rescue can be long-lasting as long as 7 days. And these persistent and bidirectional effect suggests that there might be neuroplasticity mechanisms involved, but that still leaves an open question, which is how precisely and these neuroplastic change occur and how that facilitate rule shift learning, which will be today's focus. And so for this paper, we didn't use any new technology or use like any new method, but basically we use a rule shifting and a rule reversal task to assess different type of cognitive flexibility, and we use in vivo optogenetics and slice electrophysiology to evaluate how what is the synaptic change of the closal PV synapse after the animal learned to utilize different different flexible learning paradigms. So I will go quick on the methodology. So basically, our version of the rule shift and the rule reversal learning task are adapted from the classic attentional set shifting task, but we do it in the animal's home cage. So basically, in each trial, the mice will face two balls. They are scented with one of the odor, and they are and a food reward is buried in a scented media in either sand or cat litter. And in each trial, food deprived habituated mice are presented with these two balls, but only one of them contains a food reward, which is a peanut butter chip. In the initial association phase, the animal need to learn to associate one specific order or texture cue to get the reward. For example, the animal need to attend to odor one as a rule to get the reward. Once a learning criterion is learned, which is 8 correct trials out of the most recent 10 trials, the rule will next transition to either rule reversal or rule shift. In rule reversal, for example, here, the older one no longer predicts the reward, so that now the animal needs to relearn to attend to older two to get the buried reward. On the contrary, we can also let animal to learn the rule shift. In this scenario, not only the older one doesn't predict the food reward, the animal now need to attend to a completely new dimension of cues, so that now the animal need to completely disregard of odor and attend the texture to get the reward. In order to learn this new association, mice need to go through another trial and error period, and reasonably they will make errors and also have an increased number of trials to reach the new learning criterion. So in the rule shift trials, the animal can face two different scenarios. So in the conflicting trial, which is here, the animal need to completely abandon the older one associated rule and attend to a new and attend to texture A. However, there are also trials the animal can still follow older one and get it and randomly get it correct. And this is a low effort trials, and this is a non-conflict trial. When in the conflict trials, when animal make errors, it's called a preservative error, and this is the main indicator of cognitive inflexibility. Okay, so
Just
also
Xiyu, just to double check. So on each trial, all the 4 stimuli, the 2 odors and the 2 textures are presented. So each trial involves all of them.
yes, each
But
trial all
correct,
4
but right, but which one is associated with reward depends on the rule. And then the rule change, whether reversal or shift happens within a session, right? So they start with one rule and then unknown to the rat, it transitions to another rule.
Yeah, unknown to the mice, they need to infer through trial.
Mice, right? These are mice.
Yeah, these are mice.
Oh wow, okay.
And that's why we are surprised, like they can even learn
it. I'm also very surprised. And then this happens. So each session had both rule reversal and rule shift.
Yes.
Okay, thanks. But that's
Uh,
very impressive. I guess
uh-huh.
it's odors and textures. These are salient stimuli
Yeah.
for them.
And we think it is the home cage testing
Hmm,
that can help with the
I see.
habituation.
Hmm,
Uh, yes, and also this task can be repeated on several days, and their performance remain relatively well. And they can learn the association typically within 15 trial-ish, and they automatically shift, I believe, within 16 to within 15 to 16 trials for most of the mice
hmm.
when we like switch the rule.
One other question, how much do you think the food deprivation compared to the typical water deprivation matters here?
So you mean like how many days?
No, I'm saying we typically most labs do water deprivation. So do you think that the food deprivation could be a factor in helping these mice
Yes,
learn?
yeah, we think so. Like, I mean, motivation needs to be there. So right, and we have tested. Our mice were controlled at 80 to 85% of body weight, and we also have tested like 85 to 90% of body weight mice, and comparatively, they learn less well compared to the our standard mice. Okay. So after the in this study, we also label long-range PV axons with channel rhodopsin. To do this, we inject the PV Cree mice in one PFC hemisphere with AAV5 diol channel rhodopsin UIFP, and then in our 2023 study, we find that in the contralateral hemisphere, we can observe fluorescent axons. And we also performed current clamp recording, and we find that not only the fluorescence is there, these axons were axon-looking structure at the time actually form synapse onto contralateral pyramidal neurons. And we find that most of the connected neurons are projecting to medial dorsal thalamus. However, another subset of cell of connected pyramidal neuron in the contralateral hemisphere actually projected to dorsal striatum. Other pyramidal neuron subtypes in the PFC in the contralateral hemisphere does not seem to be connected. So we conclude that the medial dorsal thalamus and the dorsal striatum are the primary target of callosal PV output. So in this study, after we labeled the callosal PV axons in the PFC after the behavioral paradigm, and we recorded in the contralateral hemisphere, and we searched for connected pyramidal neurons. The connection were identified via whole cell recording of optogenetically evoked iPSC, and we use a surge pulse of 2.5 milliwatts of blue LED pulses, and they are delivered every 30 seconds, if not longer. And also in a subset of the connected neuron, we also assess their short-term plasticity via parapulse ratio recording, and the interstimulus interval is at different intervals ranging from 1,000 to 50 milliseconds. So we in this study, like I mentioned, we also labeled PFC MD neurons to improve the recording yield, because blindly search for the connected neurons is a very painstaking approach, and the connection probability in all the neurons is quite low, less than 10%. Therefore, we use AAV retro CAG TD tomato injected in the medial dorsal thalamus to improve our recording yield. And this PFC MD loop also play an important role in rule shifting behavior, which I will delve into more deeply in the discussion session. So here are the results of this study. So in the first experiments, we want to learn whether rule shifting learning itself can strengthen closal PV synapse. In this experiment, like I explained in the methodology slide, we injected PV mice with AAV diol channel rhodopsin E1FP in one PFC hemisphere, and we retrogradely labeled PFC MD neurons. After 7 to 11 weeks of virus transduction, we performed behavioral testing in the behavioral paradigms. The animal, once they learn the initial association, they need to either relearn, or they need to either continue to learn the same rule, or they need to learn the reverse rule, or they need to learn the shifted rule. Behaviorally, as expected, animals spend more trials learning the new rule comparing to continue to learn the IA rule, and also animal make more errors during the respective learning process. Next, within 24 hours of the behavioral testing, we recorded the closal PV synapse using blue LED pulses. And we find that after around 8 weeks of virus expression, we can reliably identify axon-looking structure in the contralateral hemisphere. And they are around PFC MD neurons or other neurons of interest. And by shedding 5 milliseconds of blue LED pulses, we can find time-locked activity around the light pulses. And also, they appear to have a parapulse depression when there are two adjacent pulses delivered at a short interval. So here we looked at how a different type of rule shift learning can affect closal PV connection strength in PFC MD neurons and unlabeled adjacent neighboring neurons. We find that in the PFC MD neurons that are TDT positive, differential rule learning did not affect the detectability of those cells. However, in the connected neurons, we find that only the IARS group increases the connection strength in the contralateral PFC to MMD projecting neurons. Also, in a subset of the connected PFC MD neurons, we followed up with PPR, the parapulse ratio recording, and we find that these neurons have a decreased parapulse ratio, suggestive of increased parapulse depression, and indicating they have a stronger short-term plasticity related to presynaptic release probability. However, in the unlabeled neurons, which are TDT positive, we did not observe this effect. In fact, these neurons are less connected. And however, in the connected neurons, we did not find any difference in the connection strength or the short-term plasticity when we deliver parapulse. We realized this, the N is quite low in our initial experiment. We next follow up with a new
questions you. So the amplitudes that you measure, are they measured during the stimulus presentation? At what time point during the trial?
So these are so these are after the trials. So basically, animal went through rule shift learning, and
Okay,
we sacrifice the mice within 24
I
hours.
see. And then this is slice. So the old slice, I see. This is not in vivo
Yeah, so these
report.
are the physiology data.
But then
And
I thought you said each session had both rule reversal and rule shifting.
oh,
So
yeah, I apologize. I mean each session has IA, and then after the IA, the mice went into like different
I
learning.
see. Neither, right. What is IA again? It's the
So I
initial
is
association. Yeah, yeah.
the it is
Okay,
the initial association rule. So they
okay.
repeat the same amount of rule.
Yep,
Yeah,
yep,
or
yep.
yeah, are
And then, so after one session of either the initial association or
right, right.
switching to reversal or shifting, you sacrifice the mice and then you do this slice physiology to figure out the
Within
synaptic connections.
Right. So we chose this time window because in our 2023 paper or in all of our previous papers, we find the behavioral change is long lasting
Mm.
at a minimum of 24 hours. So we are confident that the observed plastic change uh, is at a synaptic
And
level.
this is after a single exposure to a rule shifting
Yes, this
paradigm.
is after a single exposure to rule shifting.
Right. Okay. Thank.
Yeah. So Uh, right. So yeah, in the next experiment, we increase the power. Like I mentioned, the medial dorsal thalamus and the dorsal striatum are the largest two populations receiving closal PV input. Based on our tracing data in 2023. Therefore, to further increase our yield for the other neurons that are non-PFC MD, our newest experiment, we injected a CTB to retrogradely label PFC MD neurons and PFC dorsal striatum neurons in the contralateral PFC, and then we perform the same learning task, and also we perform the same patching experiment, and we find that indeed in other cells including PFC dorsal striatum neurons and other sparse connection, there are no change after differential rule learning. This suggests that the observed plasticity is specific to closal PV to PFC MD neuron connections. We also recorded spontaneous synaptic activity of the connected PFC MD neurons, and we did not find any change in the spontaneous iPSC and spontaneous EPSC, suggesting that these neurons are not suggesting that the plasticity we observe is specific to closal PV rather than globally changing the inhibitory or the excitatory tone onto the connected targets. So after figure one, or after the first experiment, we now know that in normal circumstances, rule shift learning compared to rule reversal or continue to learn the initial association specifically induce plasticity in the closal PV synapse and in the closal PV synapse onto PFC MD neurons. Next, we want to see whether closal PV terminal activity is actually important for this plasticity, and also can this alter the rule shifting performance as observed like before. So in this experiment, we injected one hemisphere in the PFC with a mixed virus. So all the animals receive a diol channel rhodopsin to label long-range PV terminals. However, half the mice will also receive a halo rhodopsin in the PV neurons and to allow for halo to express in the axon terminals. And the other half of the mice receive a control virus as the opsin control. In the contralateral hemisphere, the animal as before receive a retrograde labeling of PFC-MD neurons, and we implanted an optic fibers to manipulate the callosal PV axons during behavior. After 7 to 10 weeks of virus transduction, we during the behavior, we deliver light during the rule shifting phase. Once the animal learns the initial association, and we measure the behavioral performance during the rule shifting behavior, and within 24 hours, we sacrificed the animal, and then we recorded the callosal PV connection strength and short-term plasticity. So here is their behavioral performance. As that demonstrated in our previous paper, we find that by inhibiting only the callosal PV projections, the animals require more trials to reach the new learning criteria in rule shifting trials, and also the animal make more preservative error along the process as predicted. Next, within 24 hours, we recorded from the contralateral PFC just beneath the optical implant. In the region that contains callosal PV fibers that expressing UIFP, we recorded both PFC-MD neurons that are TDT positive and also neighboring unlabeled neurons. So basically what we find is that in the PFC-MD neurons, inhibiting callosal PV terminals during behavior, they reduce the connection strength, and also it impairs the pair-pause ratio. However, in the unlabeled neurons, these effects were not observed. Here is a quantification of that. Again, in these animals that receiving rule shifting, receiving a callosal PV inhibition during rule shift, there is no change in the detectability of the neuron. However, in the connected PFC-MD neurons, we find decreased connection strength, and we find impaired pair-pause depression in the connected PFC-MD neurons. In the unlabeled pyramidal neurons, next to the PFC-MD neurons, however, we did not find this effect. Interestingly, we also repeated the exact experiments, but changing the behavioral paradigm from rule shifting to rule reversal. And when we find that if you inhibit the callosal PV interneuron, if you inhibit callosal PV terminals during rule reversal, there is no change in these synapses, and there's no change in their amplitude of connection. There's also no change in the pair-pause ratio recording. This further suggests that it is the callosal PV activity during the rule shifting that's important for the potentiation of these synapses, and also for the behavioral, for the cognitive flexibility behavior. So next, in the third experiment, we attempted to see whether we can rescue the behavioral phenotype by by artificially introducing callosal PV activity at a gamma frequency. So in this experiment, we followed the exact surgery paradigm and labeled callosal PV axon with channel rhodopsin and also halo rhodopsin. However, in the behavioral testing, the animal on the first day, they receive callosal PV inhibition, And on the next day, half of the animal receive 40 hertz stimulation of the closal terminals, and the other half did not receive any light. Within 24 hours, we sacrificed the mice and we patched from the connected PFC MD neurons and other targets.
So the 40 hertz pulses. So this is done through optogenetics.
Yes. So the behavior here was done in vivo. So basically on day one, they learn the rule shifting with closal PV inhibited, and on day two, half of the animals were stimulated in the same pathway during the behavior, during a second behavior.
So the stimulation, the 40 hertz pulses happen when there is no opto activation or inhibition. So the opto activation and inhibition happen on day one.
Yes. So the 40 hertz stimulation, right.
Okay, so your day one is just constant stimulation of the
halo, yeah.
Right, and you do the 40 hertz pulses through the channel rhodopsin.
What's that? Sorry.
Yeah, so if I thought initially you showed us that constant channel rhodopsin activation also impairs the behavior.
Oh, sorry. So the second figure is constant inhibition. Like
Yes, but was your figure one not about constant activation of the channel rhodopsin, and you showed us that there is some behavioral impairments?
Oh, no. So figure one doesn't have any in vivo manipulation. So
Here. Okay. So this
yeah,
is, there is no optogenetics here.
right. So the optogenetics here is a slice optogenetics.
Okay.
So we need to apply a search pause during the to search for the connection. But during the
I understand. Yeah, thanks.
Yeah, no light at all. But in the third experiment, during the behavior, we give animal. So on day one, we inhibited closal PV circuits, and on day two, they are either rescued. They are either stimulated at 40 hertz or received no light. Their day one performance, as expected, shows in cognitive flexibility impairment. They spend longer trials. They spend longer time to reach the learning criteria, and then they make more preservative errors. However, on day two, in the animals that did not receive any light, their rule shifting performance remained to be poor. However, in the animals that's actually receiving 40 hertz pulse of the closal PV projections, their abnormal behavior is rescued, as demonstrated here and here.
Okay, I feel, but don't we need to have a control where day one did not have the inhibition and day two did have the 40 hertz pulses? So I'm saying, what is the effect of these 40 hertz pulses on their own without a prior opto inhibition?
Yeah, that's a very important question. Yes, so these are control experiments that we did, but it's not included in this paper. But we find that in the animals that does not have any previous exposure of like 40 exposure to red light, just exciting those terminals at 40 hertz does not induce any synaptic potentiation or plasticity.
And it doesn't change the behavior either?
It doesn't change the behavior either, and the animals still remain quite flexible.
Okay. One other quick technical question. How long do you do this opto inhibition? How long you do it throughout the entire trial?
Yes, we do it throughout the entire trial, and also in our no light control group, we also which is not like included in this paper. We also match that amount of light exposure to.
But then we're talking about several seconds of constant opto inhibition of PV.
Sorry, for the constant halo, it's about 10 to 20 minutes. And for but like it is at like a low power. Like it
But
is
20 minutes, so you do it throughout the session. It's not like you do it one trial, then no inhibition during inter-trial. So for 20 minutes, constantly you're shining light.
yes, for the halo. But like for the pulse stimulation, yeah, we are doing it. Yeah, we are doing it at 40 hertz. So the light power is presumably lower.
And have you measured the activity of these neurons? See what 20 minutes of constant upper inhibition does to them?
So
That's a little bit too extreme to for such a long time do this. So that should really change the activity of these neurons. Like I don't know what it does with
um
brain.
yeah, so I think we have done two follow-up experiments. So first of all, like we have done so inhibition or stimulation just when the animal can commit an error, and we can fully replicate the behavioral effect and also fully replicate the phase effect in our newest cohort. So indicating that the heating effect is not driving for the observed effect here. And we also have done um, uh, exposure to long light without any option. And we,
Yeah, so what I didn't mean heating effects. I just mean the effect on the electrophysiological properties of the cell that the neurons will be really pushed outside their standard EFIS properties with such a long.
yeah, I think that could be a caveat. Yes.
Okay.
But yeah, so but since we can replicate it with a post outcome inhibition, we believe that like we believe that like this, but like it's a pilot data set. We plan to add to this, uh,
Okay. Sure.
and we still need to follow up with more animals.
Sure.
But we believe that with just like a post outcome inhibition, to timely intervene with activity patterns, we can replicate the behavior and also the EFIS effect. But like that's up to a more n to be added to that data set. Right. Okay. So electrophysiologically, we find that so here is in the PFC MD neurons. Oh, I'm also checking the time. Okay. So I will wrap up more quickly. Okay, so basically in the PFC MD neurons, we find that the behavioral rescue is associated with also rescuing of the electrophysiological effect. In the percent of the cells in the in the connectivity, close to PV connectivity, we find that two inhibiting closed PV terminals introduced a lower connection probability, and this effect is is rescued by subsequent 40 hertz stimulation of the same projection. We find that there is also a deficit in their connection strength, and this is also and this closed PV inhibition associated with OFSC deficit is also rescuable when we excite the terminals at gamma frequency. Surprisingly, we did not find any short-term plasticity deficit when we tested animal twice. But since rule shifting itself introduced plasticity, we postulate that this could be due to repeated testing, blocking the repeating testing itself introducing plasticity. Again, in the other in the unlabeled in the non-PFC MD neurons, we did not observe any change in the connection probability, in the connection strength, and in the short-term plasticity of the closed PV synapse. So this leads us to the discussion, and here's a summary of the findings. So basically, through this study, we find that indeed, rule shift learning itself can potentiate long-range PV synapse in the PFC, and we find that those long-range closed PV synapse they project onto cortical thalamic neurons, and they are selectively potentiated by rule shifting, but not by rule reversal or by continued learning of initial association. This suggests that the closal PV synaptic strengthening in the PFC is experience dependent. And also, we find most of the described effect happen in the PFC to MD neurons, but not other unlabeled neighboring neurons. And this suggests that this type of synaptic plasticity is cell type specific. And also, we find that by manipulating synaptic activities of the closal PV synapse, we can prevent learning and induce a long-lasting preservative state. And we also find that by this preservative state can be rescued by subsequent closal PV stimulation at a gamma frequency, suggesting that the observed synaptic plasticity and also behavioral changes are.
Did you try the other frequency ranges to see if it is specific to gamma, the rescue?
We haven't tried for the patching, but in our 2023 paper, we tried to rescue. In our 2015 paper, we in the animal model with impaired rule shifting performance, 20 hertz stimulation, 20 hertz in-phase stimulation of both left and right hemisphere PV does not rescue their rule shifting behavior. But in terms of the synaptic plasticity, we don't know. Yeah, but that remain a future directions we plan to test to see whether these are frequency dependent. Yeah. Also, here is the strength and the weakness we think of the paper. So first of all, we looked at a circuit that is novel to rule shift learning and also to the PFC. And we find that the long-range PV synapse in the PFC, they are indeed plastic rather than static. And also, this paper, we focus on PV output synapses, whereas a previous description of PV where inhibitory plasticity focused on the input synapse onto GABAergic interneurons in the PFC, which we believe is a conceptual innovation of the paper. And also, we in this paper, we use optogenetics behavior and also patch clamp recording all from the same mice. And we believe this multimodal technique can lend us with more confidence in the observed effect. However, we do realize this paper is limited because patch clamp technique is a terminal technique. When does closal PV synapse potentiate during the behavior? Does that correspond when rule suddenly change? Is still unknown. However, we plan to test it with silicon probe recording in our next project. And another conceptual limitation for this paper is that although we know that the rule shifting induce plasticity in this long-range PV circuit, we don't know whether other flexible learning, such as the learning in the T-maze, where whether spatial flexible learning can also trigger the same or similar plasticity mechanism. And also in the study, we did not test for local synapse, and also we did not compare directly whether local synapse also exhibit plastic change during this process. However, we will address that in a future study, which we have already started. So we believe that the observed plasticity can contribute in vivo to cognitive flexibility, potentially in two ways. First, we speculate that the strengthened closed PV synapse may directly connect to the ensemble neurons associated with the IA rule. Therefore, potentiation of those closed PV inhibition can suppress the previously learned rule representation, and thereby facilitating formation of the new rule related ensembles. Another possible model is that the strengthened closed PV synapse may synapse broadly onto all the PFC MD neurons, since the PFC MD loop is known to serve to monitor the behavioral feedback. This could lead the animal to have trouble or to have difficulties to maintain the previous rule, and therefore forming other new rule association. But perhaps other hypotheses can also explain this. Maybe the closed PV neurons can also themselves form ensembles and also introduce neuromodulation to affect neuromodulatory effect and control motivation, control motivational state change that affects PFC circuit that's important for rule association learning or cognitive flexibility. All these possibilities, I think, could stand, and we are currently using in vivo techniques, silicon probe recording to test them one by one. And also for future directions, so like I mentioned, we plan to also study whether local PV neurons can have similar plastic change. And we also would like to understand the molecular mechanisms involved in the observed plasticity to see whether it involves different GABA receptor subunit and whether it require an MDA related mechanism. And also we are also interested in whether the observed synaptic potentiation is involved in animal model related to schizophrenia and right now and other neurodevelopmental disorders. Right now we have some preliminary evidence suggesting that it is not an artificial effect triggered by our optogenetic manipulations. Indeed, in various animal models of schizophrenia and related disorders, we did find reduced closed PV plasticity associated with their perseverative behavior. With that, I want to thank the opportunity to present my work here. I also want to thank my PI for giving me this opportunity to study this projection. And this study is supported by two graduate students in the lab, Lara Hagelpin and Kira Walquist. I also thank our technicians, Lina Shindi and Tegan Balak, for the technical support. With that, yeah, thank you very much.
Thank you. That was very well presented. I don't know if